Hello.
You know that soap
Mhm.
that we made?
Mhm.
I left mine in my locker,
Uh-huh.
and now it smells awful.
Okay, so we're going to start today talking about some peptides. Just as a review, what is a peptide linkage? It's amine. So we have the amine group that's going to attach to the acid group, C, and we get this linkage, right? Okay, with it coming off like that. Okay, now um, we're going to be talking about today small peptide linkages. And when we talk about small peptide linkages, we're looking at about 5 peptide linkages. And these have to do with hormones. Okay? Um, as we look at the hormones, there's basically 3 different ones. We're on page 348. 3 different ones that we want to be worried about. So this is small linkages. We're talking about 5 of them. And we've got, and I am not going to try and say these out loud because I'll probably screw up.
Wow, it's like an exam.
Okay, and this one right here is TYRGLYGLYFE met. And this one right here is TYRGLYGLYPHELEU. Okay. So as we look at these 2 right here, the bottom 2, these 2 right here are neurotransmitters. What can you see that's the difference between the 2? They're both really close. The very end, the met and the loose. These have to do with how we deal with pain, okay, and how our body reports pain to us. The first ones, the E ones, are pentapeptides that are neurotransmitters produced by the brain itself to bind at brain sites. The last 2 right here are pain reducing effects. They help us to reduce that. So let's go on to the next one. I just want you to know that there's 3 and this is what they're doing. We're not going to go really deep into this. We're going to go now to the small peptide antioxidants. And on the page 348 at the bottom, there is a drawing that is glu-cis-gly. You see it? Okay. This drawing here is of a tripeptide glutathione. I cannot even say that. Okay. And it helps protect from oxidizing agents and peroxide. So it's like the helper for skin not being oxidized and stuff like that. All right, general structure of proteins. So we're going on to when we talk about general structure of proteins, we're going to be talking, they basically have at least 40 amino acids that are put together. And if you look at the table on the page 349, let's look at that together. I'm going to put it up. I'll move it here in a minute so you can see it. That's not going to show real great, but that's okay. We'll just take it and move it down here. Okay, so this is our table that's on that page. Now, with that table, we want to look at, I'm going to pull this up now so I can see it, some things that are going on there, some specific examples. And we're going to go into these specific examples right here later on in the chapter. So know that we're going to be working with these different kinds of proteins and what they do coming up. Okay? So this is just a good little overall version look at. Okay. So we already know that a general structure of a protein, basically on one end we're going to get NH3+, we're going to have a long chain here. On this other end we're going to end up with COOH and I have a minus there. Why do I have a minus there? Yeah, if I do COO and a minus, then we have basic chain here, and then the linkages come by how they go together. So they're the peptide linkages. Okay, because of this, and I am now in 910, so we're really flipping through here. Because of this, and you can this can be any amino acid sequence in here. So you've got your amino acid sequence, and it could be, you know, any amino acid sequence. But we could basically end up with a protein that has an amino end and has an acid end. Now, because of this, when we make peptide linkages, when we put those linkages together, it actually gives us a cis and a trans that are possible because we have a rigid structure. Okay, so we can do cis and trans within this structure right here. Okay. We also end up doing in what's called secondary structures of proteins. We end up having two main structures. There's the alpha helix, and there is the beta planar, I think it's what it's called, pleated sheet. Pleated sheet. These have to do with these long chains that we've got up here, and sometimes those chains will wrap along each other and they'll connect. Maybe they'll have a bond that's sort of connecting it here and here or down farther. So this is an alpha helix. So this one is more like the alpha helix. A pleated one actually comes and it wraps on itself like this. And the bonds form between like this. Okay? So it becomes a sheet. Hence why they call it a pleated sheet, because it looks like it's pleated. And an alpha helix, I always think DNA when I think alpha helix because it's a swirly or corkscrew kind of thing. Okay, now I've got a little note to look at the bottom of page 352 and the top of page 353. They've got fancier drawings than what I just drew up here, but they give you the same idea. Oh, sorry, I fell off my shoes. I stubbed my toe real bad last night and broke it, so. Okay, so when we look at secondary, we're thinking alpha helix or beta pleated helix. Now the whole chain doesn't have to be all in helix or all in pleated. You can actually have both of them intermixed at different parts of it. So you can have a chain going along like this, and then it does a helix, and going along, and then it does a pleated, and then going along, and maybe it does another pleated, maybe going along, and it does another helix. So it can keep going like that. Okay? But one end has to be your ammonia, your amine, and one end has to be your acid. Okay? All right, so as we look at this, we actually have, so that's a secondary structure. This would be like a primary structure up here. Primary is just like a straight line, primary. Secondary structure, we've got these two right here, okay? And this right here, what I've drawn over here, is a tertiary. Did I spell tertiary right? Okay, this is a tertiary structure over here, which intermix those other two, okay? So if I have a simple primary, it's just a straight line. If I have a secondary structure, it's either just alpha helix or it's a beta pleated sheet. Tertiary mixes all of it. Okay, so how do I get these things to happen? Well, these bonds right here that I was talking about, here and here and here and here, any bond in here, have to be one of four kinds of bonds. So let's talk about those four types of bonds. Actually, this is more with a tertiary than any of the others. Okay. When you're looking at that, this all has to do with the R groups. Okay, remember talking about the R groups, how some of them were acid, some of them were polar, some were nonpolar, some were basic, that kind of thing. With this, when we talk about the R groups, if we have covalent disulfide bonds, so it's reached across and it's got a sulfur to a sulfur bond. Okay. It could be electrostatic. What is electrostatic? Anybody remember? I always think electrostatic like cling-free. I need cling-free because I've got static electricity on my stuff and everything wants to be collected on it. Okay, so electrostatic attraction, I've got a plus and a minus and they're attracted to bond. Okay, I have hydrogen bonding occurring. And a hydrogen bond is between a nitrogen and a hydrogen, oxygen and a hydrogen, or a fluorine and a hydrogen. And we usually don't have the fluorine and the hydrogen in organic. And then we also have hydrophobic. What is that? You have a phobia of hydrogens? Close. Attractions. This is where hydrophobic just means it doesn't like water. Okay? So if I have one end that likes water, then I'm going to have one end that doesn't like water. So remember when we saw these, when they did this, and we had one end that was water loving and the other end didn't like water. We'll do it like this. Okay? So they arrange according to who they like, and this is where we get the membranes. Remember that? Okay. So there is even one more past this. It's called a quaternary, which means what? For protein structure. This is what we call multimeric protein in its organization of peptide strands. It has even number of dimers, which means it usually has 2 or 4, something even. An example would be a hemoglobin. Globin, I can spell really. Okay, so let's look at a picture of a hemoglobin on page 358 of your books. Okay, I'll try and bring the picture up here. Bring it down. Okay, so this is a hemoglobin, and notice it has 4 different groups. Now, as we're looking at this, we have a beta chain here. We have a beta chain here. That's 2. And then we have 2 that are alpha chains down here. Okay, so there are even amounts of that. So this is what we call a quaternary structure. Now, I really like how this book will give you chemistry at a glance, because we can now take and review real quickly everything we were supposed to have learned. Okay? And this didn't project really great, but primary structures, a sequence of amino acids. Secondary structures, remember we've got the alpha and we've got the pleated sheet, the alpha helix and the pleated sheet. Ternary structure can be a mix of the pleated sheet, the alpha, and the primary.
It's like a snake stick.
Sort of, yeah. And then the quaternary, we have at least two of the same thing around something that interconnect, okay? All right, so now we're going to page 250. Where have I got? Let's see. 358. I already did chem at a glance. Okay, so let's talk about the classification based on shape, because this becomes really important. So as we go to classification based on shape, we basically have two main groups. Okay, one is called fibrous. Did I spell that right? Yes. Protein, and the other one is globular. And I know I'm not. Okay, so as we look at this, globular and fibrous, there's a couple things that we need to look at. This fibrous is usually long, like fibers, okay, intertwined like this. Globular is just like what it says. It will interact and be a glob, okay? These are generally not water soluble, so let's say insoluble in water. So we can do the little cross through like that. Doesn't like H2O. This one over here dissolves readily in water. Okay. And that's because the side that's out is the water-loving side. All right. Fibrous proteins are usually used in structural support, like our muscles and our bones and stuff like that. Whereas globular are used to transport things, okay, in our system. We are mostly made up of fibrous proteins. So let's look at some specific ones. Alpha ketrin, we're almost done for today as far as notes, so stick with me, we're almost there. Let's do this. Alpha keratin. This is a fibrous protein, and it is for used as protective coating. And you can think skin, nails, like fingernails, toenails, feathers, wool, claws, okay, stuff like that. And it is alpha helical, which means it's going to be that spring shape, okay? Also fibrous is collagen. I know a lot of people, my age anyway, think, oh, I got to get some collagen injected. I've got too many wrinkles. Okay, so collagen is the most abundant in humans. It's 30% is what our body, of all the proteins we have, that's how much is that. And it is tendons. It is your ligaments. It is the blood vessels, ligaments. Skin, sorry, I said skin was under alpha keratin, but it's really nails. And bones and teeth have a lot of collagen, though they're not totally. This one is a triple helix. So it gets a little more complex. To get a good picture of that, on the top of page 361, you get a good look at what that looks like right here. Okay, so you can see that it actually intertwines, and you're looking at a good picture right here. Okay? All right, next one you've probably heard of a lot of, is hemoglobin. What does hemoglobin do? Why is it so important? It transports oxygen. And it transports oxygen from the lungs to the rest of the body around the room. Okay, so to the tissue. It is ternary, and it has 4 peptide chains, and we looked at a picture of it earlier. Each subgroup within this ternary molecule, remember we said there were 2 beta and there were 2 alpha chains, remember that? And we did them in green here, and we did, I think the bottom one's in blue like this, okay? Each of these areas right here are known as a hem, okay, or hem group. Okay, and what happens here is in the center, we have an Fe+2, and that's what interacts with oxygen. And the last one that we need to talk about today is myoglobin. And this is what, when the oxygen gets to your tissues in your body by the hemoglobin taking it there, the myoglobin actually stores the oxygen in your muscle. Okay? And it is also ternary. And it has 1 peptide chain and a hem monomer. Okay. All right, we're going to do a little activity, and you're going to need to do open up your book to page, where is it? Oh man, come on. To page 362, to the chemical connection. And the chemical connection there is protein structure and the color of meat. And what I would like you to do, and we're going to talk about the activity that we're going to do here in just a second, in connection to this and to the rest of the chapter as we talk about the rest of the chapter next time. Okay? So before you do that, I do want you to read the chemical connection and sort of write down what things you didn't know about that beforehand. But I want to also make sure that you have your assignment, which is page 375, numbers 67, 71, 72, 73, 74, 75, 79, 81, 85, 89, 90, and 92. Okay? So we'll be working on that while we do a little activity. Now what we're going to do is I want you to read this activity, this chemical connections, and write a little bit about it into your notes. Then we are actually going to observe hamburgers being cooked and then eat them. Okay? So, now.
Really?
Yeah. So I'll get that going. You guys read this chemical connection, then we'll come back together and talk about it, and then we'll do that little activity, and you can work on doing your problems. Okay?